I would like to invite the next speaker, Dr. Amit Ghosh from IIT Kharagpur. So he works on fulfilling the dream of a sustainable planet with respect to renewable energy. And in today's talk, he'll allude to his work on systems and synthetic biology for microbial production of biofuels and chemicals.
Thank you, Arijit, for inviting me. It was so far a nice conference, and it was nice hospitality and hard work from Arijit for organizing this event. So I will be talking about application in energy. So far we have a lot of talk on health, different kind of disease. So the modeling in the first, I will talk about the genome scale model, how genome scale model can be used for characterizing different organisms, and how you can use that potentially for producing different biofuel and chemicals, which has commercial application. So I divided my talk into these three sections. So especially if you see that biofuel, the energy comes from fossil fuel, right? So there's a lot of carbon deposition in the atmosphere. The more you consume the fossil fuel, it will be harmful, because more carbon dioxide and more emission will be there in the atmosphere. So how you can replace the existing fuel with a renewable fuel that can be regenerated, right? Also there you have to see about the emission. So the biofuel are, if you see, there are different generation of biofuel. Basically this is the first generation where food is used. Sugarcane juice is used to produce bioethanol, and then USA produce bioethanol from corn. Whereas in India and China, specifically, we focus on the lignocellulosic biomass because of the constraint from the government that food cannot be used. So this the entire plan is basically how you can utilize the lignocellulosic biomass, which is a non-food part of the plant. And these require a lot of challenges. For example, you have to pre-treat the biomass so that you can release the sugar from the lignocellulosic biomass, and then you have to hydrolyze and then ferment. So mainly we work on the pre-treatment area, and then also in the fermentation. So as you know, the biomass which is in the form of a complex sugar like cellulose, hemicellulose, and lignin, we generally use ionic liquid to actually decompose the lignocellulosic biomass. So here we also do, I will not be talking about this thing here today. Especially we apply ionic liquid and do molecular dynamic simulation to identify what concentration of ionic liquid and what kind of mutation you can do in the enzyme, so that it can accommodate the ionic liquid. So it will be a one-pot process where ionic liquid will dissolve the lignocellulosic biomass. At the same time, the ionic liquid will not harm the enzyme, because enzyme will also be tolerant with the ionic liquid. And then lastly, we have to ferment the sugar to get the biofuel. But this process required a lot of technological development to improve its production. So if you see some of the products which are on the market, for example, the artemisinin, which is produced from Saccharomyces cerevisiae, and the project went for 10 years, and the cost of the project is basically $50 million. And then similar product was also developed by DuPont, which making butanediol, and that project also ran for 15 years, and the project cost around $130 million. So these challenges are more, and there is a long-term challenge that we achieve through long-term project. So recently you can see the video which was produced by Genomatica that was open in 2016 also involved the systems and synthetic biology, how we can use effectively to produce this compound. This is used mainly in the plastic, and right now they produce 30,000 tons of BDO every year, and which employs 70 people, and the production unit is in Italy. So these are the success story I told you, but there are a lot of products which are actually in the lab. So it's not because the production level is not that good. So this is the cycle we follow, that build, design, test, learn. So this is what we construct the pathway. So it involves synthetic biology informatics. So it's another computational branch where you design a pathway with a particular set of promoter, and then want to see that how you can integrate the different biological part in the chromosome. So as you know that because of the revolution in genome engineering like CRISPR-Cas9 and other genome editing tool, nowadays you can effectively integrate any amount of DNA in the chromosome so that you can have more enzyme inside the cell. And then you have to identify that because of this modification in the genome, whether there is any problem in terms of through high throughput sequencing and other technique, we would be able to test whether the organism is able to produce enough amount of the product. And then you also do flux analysis, metabolic flux analysis, kinetic modeling, and then machine learning model is also recently people are using. So machine learning models are basically are not mechanistic model. So this is also used to identify what are the bottlenecks in the metabolic pathways. So this is one example in the machine learning model that people generally apply, that you have the expression like this is a small metabolic pathway which has 2 genes. And we want to know what should be the expression of these 2 enzymes such that you get your product more. So you can simply construct a decision tree and see that if the protein expression lies between this range and also lies between this greater than Y1 and then P1 greater than X1 and less than X2, then only you see your product is maximum. So this kind of decision trees you can make from the existing data. So right now you will see that a lot of data or database exists today which store this information. So how different enzymes are expressed and how you can improve the production. You can also construct the machine learning model to improve certain product. So then I will move into, apart from machine learning model, the mechanistic model is basically what Dr. Anu has explained, the genome scale model. That is extensively used in bioengineering, where you construct the genome scale model using the annotated genome. Suppose you have come across a very important bug that can produce higher amount of certain biofuel or product. You can immediately sequence that organism and annotate the genome using a lot of annotation server exists today. You can identify which are the protein which are catalyzing the reaction that you can identify and then build a stoichiometric matrix. So Anu has already explained the stoichiometric matrix. The stoichiometric matrix you can use in calculating the metabolic fluxes. So the fluxes are actually the rate at which the molecules are moving from one metabolite to the other. So this actually help you to understand that how the carbons are flowing. And using the stoichiometric matrix, you can maximize an objective function. For example, we use the biomass equation. Since we use the microbe, you can still use the biomass as the objective function. Unlike in mammalian cell where you have to do flux sampling. But here biomass maximization is enough to get the metabolic fluxes. And from the flux, you can see what are the how the carbons are flowing, how you can improve the flow of carbon in particular direction. By this way, you can improve the production. So this on top of every reaction, in the genome scale network, we have this gene mapping. So these are known as GPR relationship, gene-protein reaction relationship. So using this information, you can easily modify the genome and try to see whether you can remove the reaction. So reactions are removed by perturbing the gene. So depending on the number of genes which are involved in the reaction, you see how you can either remove that reaction or you can also upregulate that reaction. So these are generally done in metabolic engineering, but just by looking into the genome scale model and using the Boolean relationship. So in the genome scale model, you will see that the metabolic reactions are there, but also these reactions are mapped to certain Boolean relations of genes. So these are used effectively for engineering. And then the genome scale model have a wide range of fluxes. So this, when you solve this equation by applying steady state condition like S dot V equal to 0, the solution lies in the null space. So the box shown over here, every point is a solution. But what we do is basically you maximize the objective function such that the biomass is maximum, or you can choose any other reaction as the objective function, and try to fix your solution at a particular so you get a particular solution. In this way, you get the unique solution, or you get the range of solution for each and every fluxes. So this is usually done, and then we also develop a pipeline to how to construct the metabolic network. So using the basically we work on non-conventional organism where metabolic models are not available. So basically you get the genome sequence and then annotate the genome and do mass and charge balance for each and every reaction. And finally, you construct the biomass equation. So the biomass equation is, when you estimate the biomass equation, you have to do the experiment. Otherwise, everything is computation. You can easily get the drought model, and then ultimately you have to define the biomass equation back, because the biomass equation are the bottleneck of any metabolic network. So this require experimental doing the biomass need to be measured experimentally and require some amount of experiment. Otherwise, if you get the stoichiometric matrix, you can easily get the fluxes. So using this pipeline, we are constructing genome scale metabolic model in non-conventional is the yeast, which are actually, as you know, the yeast Saccharomyces cerevisiae is the workhorse in industry, which produce bioethanol. 100 billions of bioethanols are produced annually. Many other products are made using yeast. And among the yeast, so far Saccharomyces cerevisiae is extensively engineered. But if you go to non-conventional yeast, there are a lot of potential yeast which can produce a lot of product, and apart from bioethanol, because bioethanol is produced naturally. So this non-conventional yeast like Lactantia kluberi, we have annotated the genome and we've developed the metabolic model. Before you can publish this model, we have to validate the model using knockout, gene knockout, and try to see what is the accuracy of the model. And also, once the model is behaving very well with the experiment in terms of knockout, you can perform the phenotypic phase plan to identify how much ethanol it is produced, how much ethyl acetate is produced, and with respect to how the growth rate is affected. So this is just to cross-check how the organism behave before you can go to the experiment. So after, if you verify that the organism is very useful, then you go for engineering. That's what we do in the lab. So right now we are engineering this organism, Lactantia kluberi, for the production of fatty alcohol, also for production of bioethanol using genome engineering tool. Also, we are working on microalgae. So this is the model. This is accepted this month, where this is a microalgae which is not used for engineering. So from scratch we annotate the genome and build the metabolic network using the genome, and also validate the model, and also constructed the biomass equation from experiment. And right now we are doing the genome engineering for production of different biofuel. So apart from microorganisms which are beneficial for production of fuel or chemical, also we are looking in the microbial communities, because the microbial communities are very important today. It has a very much application in health, and also in waste removal and many other applications. That's why the metabolic flux analysis can also be done in the microbial community. So for that, we earlier developed some of the tools using 13C MFA. The 13C MFA is again the extension of FBA, where you do the experiment to measure the metabolite. So you can see that the isotope level glucose is used to grow the microbe. And as the glucose is leveled, its metabolite is also getting leveled. So based on the leveling profile, so these are the leveling profile of small peptide which are unique to the organism which are present in the community, and we can easily calculate the metabolic fluxes using the metabolite profile and try to get the information of the fluxes specific to particular organism. So also we build a different kind of network for the microbes in the gut microbiota. For example, in the insect, we started with this project with the insect because insects, as you know, these insects are very powerful in degrading the lignocellulosic biomass. So in the gut they have certain kind of community which actually dissolve the lignocellulosic biomass with a much more effective way. So we want to understand what are the interaction of these microbes. So that's why we build the interaction network, the global metabolic interaction network, how hundreds of microbes are actually interacting with each other, and the interaction is through exchange of metabolites. So this is not a genome scale network, we just an interaction network. By knowing the interaction network, we can identify what are the crucial microbes, and then we go for the genome scale model. So these are the steps which we are following. First we construct the global network, and then using the global network you identify the microbes which are crucial for lignocellulosic degradation. So and then going for the genome scale community metabolic modeling. So this as you have seen the genome scale model which are specific to one organism. Suppose you have a community where we have hundreds of microorganisms. So how do you model their metabolism? So this is even more challenging because you have to handle many microorganisms together. So this is an example where we can see that some of the like 10 metabolic models we made, and then develop the community metabolic model. And the community metabolic model, you have to define a biomass equation for the community. So for the entire community you have to define the biomass equation, and the biomass equation we derive based on the metagenomic abundance. And this way you can easily get to know what is the effect of one microbe to the other. Suppose I add one microbe and I want to see whether the P is actually contributing to the community X to what extent. So if the community metabolic assistance can be calculated for the community, and based on that you can easily get to know whether that microbe is influencing the community. And using this mathematical algorithm, we are actually doing the experiment to know how a single microbial species is influencing the community, and that helps us to design different experiment for synthetic consortia. And this also benefited by support vector index, that we are pairwise growth support vector index have been calculated to know how the two microbes are actually helping each other. So depending on that, whether it's a positive influence or a negative influence, you can easily calculate from the modeling approaches, and then you can design your experiment. Because the consortia, designing a consortia at the experimental level is quite difficult, because most of the microbial consortia they don't grow in the lab. So if you know that the pairwise interaction, that two microbes can interact in a positive sense, or it can have a community metabolic assistance, so you can select those microbes accordingly and design your experiment. So this is the work we are doing at the experimental level right now. And also we are looking for the gut microbiota in bovine. So as you know, the bovine gut is also useful for degradation of lignocellulosic biomass. So in that case also we are identifying the microbes which are influencing in the lignocellulosic biomass degradation. So you can see that most of the microbes are not influencing each other, but more than 50% of the microbes are actually interacting each other through metabolite. So then also we are looking into the gut microbiota in human. So in this case we are using the metagenomic data for diabetic patient to know how the gut microbiota is actually influencing the diabetic patient. So this is very interesting work we are right now planning to submit in this month, where we found that some of the microbes actually become for a normal patient it was found that it is consuming glucose, but in a diabetic patient the microbial consortia, rather than consuming the glucose, is actually producing the glucose. So that actually become very problematic for a diabetic patient, where the glucose level is very high, because the consortia which is present in the human gut actually not helping, rather than it is creating problem. So then the other type of model we are interested in is basically context-specific model. But yesterday we have seen that Professor Nirav actually presented some of his work on context-specific model. Here we actually build the model on SARS-CoV-2 infected lung cell. So you can see that this is the normal human bronchial cell where the number of reaction, number of metabolites is given over here, and then because of the infection you can see that the number of reaction has reduced, and then also the metabolite is also a little bit reduced, and the genes are also reduced. So these are the infected lung cell and the infected lung tissue, and then also the infected this is the normal lung tissue, and this is the infected lung tissue, and we build the model using the RNA-seq data, and also build the virus metabolic model using the data available. So combining all those things, you have made the context-specific model for the human gene, and also integrated the virus model into the human gene to get the metabolic fluxes. So using this protocol as very similar to what Nirav has presented yesterday, that using the RNA-seq data, you normalize the data, and then you integrate the normalized RNA-seq data through T-init algorithm to get the context-specific model of the human gene, and then calculate the metabolic fluxes using sampling. And from various statistical state, you identify the important reaction which are affected. So here we actually find out the enriched pathway based on the differential flux. So earlier you have seen how differential gene is expressed, but using the genome scale model you can calculate the genome scale differential flux analysis. So this is very effective to know how different metabolic pathways are affected. So using this method we found some important result in coronavirus affected lung cell, and you can see that the fatty acid biosynthesis in SARS-CoV-2 infected lung cells increased significantly. That's why most of the patients infected by SARS-CoV-2 have seen that their fatty acid synthesis has increased tremendously. But at the same time, the fatty acid degradation pathway, like that is the beta oxidation pathway, is downregulated. So this basically helps the virus to grow. It can ramp up its growth very well because of the fatty acid accumulation in the human cell, and not allowing it to degrade. So that's why the virus can grow with much more enhanced rate, and because of this because virus need the cell membrane for which are made from the fatty acid. Remaining other metabolic pathways are also in the KR pathway is downregulated, and then the block pathway is upregulated. These are also validated using the experimental data, and they are very much correlating. So these are all based on the differential flux analysis, so using the genome scale model. So this was also highlighted in Ministry of Education, that the model shows that SARS-CoV-2 alter lung cell metabolism. So lastly, I will talk about the genome engineering, where as I told you in the beginning, that the models are made, and then ultimately using the model we try to make a new type of organism which can produce certain metabolite or certain product which can help which has a high commercial value. So here you can see that because of the revolution in the genome engineering tool, like for example CRISPR-Cas9, we can actually effectively engineer most of the organism. So here we have given the list of CRISPR-Cas9 tools which are available for different yeast. And first we have developed utilize the existing CRISPR-Cas9 tool for Saccharomyces cerevisiae, and then different organisms which are available in the literature, and then you build the CRISPR-Cas9 tool for the Lackensia cluveri, which is not available. So using that tool you can actually disrupt the gene, you can delete a gene, or you can integrate the gene using defining the proper flanking region. And these actually help us to engineer the organism in an effective way. So using this tool, we are actually engineering the yeast for consumption of different sugar, because Saccharomyces cerevisiae or different yeast can only consume C5 sugar, C6 sugar. Basically, they can consume glucose but cannot consume the pentose sugar that are available in the lignocellulosic biomass. So in that case, we have actually engineered the organism by finding the locus where this gene can be integrated. So the xylose consuming pathway, which are taken from other organisms, and they have been integrated in the chromosome by locating the region where the gene can be integrated. So this you can see that we target a 20 nucleotide sequence in the chromosome and design a donor DNA in such a way that it can integrate in the chromosome in a particular locus, and you can easily check using colony PCR how they are located in the chromosome. And this we have targeted for different gene in the sense that we have integrated XR, XDH, and XKS1 to construct the heterologous metabolic pathway at the chromosome, and also verified by colony PCR to know that they have been integrated in a proper region. And then we have calculated the consumption of the xylose. Like we can see that this is the organism, the metabolic engineered organism, and this is another set of engineered organism where you can see the xylitol accumulation was more here even after inserting the heterologous pathway. And then we have integrated multiple copies of the gene in different locus such that the accumulation of intermediate product, for example xylitol over here, you can see that the xylitol accumulation, because of the accumulation of the xylitol, we don't see much consumption of the xylose. So just like in plasmid expression, you express the plasmid inside the cell, but in genome engineering, we have to keep multiple copies of the gene at different locus such that you have enough protein to be expressed inside the cell so that the accumulation of the metabolite doesn't happen. So also we look for other product, for example, the fatty alcohol production or a free fatty acid production, which all can be potentially has a commercial value. For example, fatty alcohol is used in biofuel as well as in detergent and surfactant. So for that we measure the level of free fatty acid that are produced here. So the free fatty acid level can be improved just to check how much fatty acid you can produce. So using the engineering strategy, we have measured around 780 milligrams per liter of free fatty acid using different engineering strategies. So we downregulated the competing pathway. For example, glycerol is produced and which is taking away the flux from the central metabolic pathway can also be downregulated so that more carbon goes into the acetyl coenzyme formation. And also we can take out the citrate from the TCA cycle and push this flux toward acetyl coenzyme. So this way you can improve the level of acetyl coenzyme, because acetyl coenzyme is one of the precursor molecule for free fatty acid. So using this strategy, we also calculate the 13C metabolic fluxes for to know how the carbons are flowing. So we saw that there are fluxes which goes into glutamate which consume NADPH because fatty acid productions are limited by NADPH molecules. So we have cut down the metabolic pathway which consume NADPH. And not affecting the growth. At the same time, it will not affect the growth. So we targeted different genes, for example, glutamate dehydrogenase, and then the flux which are going in fatty aldehyde formation. So those competing pathways are deleted. And since we are doing using CRISPR-Cas9, you can do n number of deletion and modification. So you can generate many types of strain. And out of that, you have to select which one is best for your engineering. So in this way, we improve the production of fatty alcohol to 1 gram per liter. And so I'm done. So this is summary. So what we learned from this, using genome scale model, how you can actually target different non-conventional organisms. For example, in microalgae or in non-conventional yeast, you can apply because not much work has been done. So using the model, you know how the potential of the organism, and then practically you apply the genome engineering tool to improve the production of different metabolites. So this is the group who works. They are working on different topics. Thank you. I will take up question.
Yeah, thank you very much, Amit. So can we have a couple of quick questions, please? Yes.
Thank you for the talk. So what I understand is the flux balance analysis assumes that the metabolites are not diffusion limited. I mean, there is no special non-homogeneity in the models. So is there a way to extend flux balance analysis to cases where spatial variation of concentrations are important?
See, the basic assumption in FBA is basically there is no metabolite accumulation. So how much it is produced is used up. So if you want to do those kind of calculation, then you have to go for non-stationary MFA. Non-stationary MFA where the metabolite accumulation or all those things, where you actually solve the differential equation. So here you just solve the linear equation, you assume that S dot B equal to 0. Why S dot B equal to 0? If the concentration is not changing with time, or then you can apply this at S dot B equal to 0. But suppose S dot B is not 0, then you have to solve the differential equation. Then whatever you are saying, then it will be taken care. Understand? So this is just an approximation. So the FBA approximation is very good when the steady state condition is applied. But if it is a non-steady state, that's exactly what you do in metabolic flux analysis in photosynthetic organisms like cyanobacteria or algae, where you can see the steady state is very difficult to achieve. So you apply, you solve the differential equation, is known as INCA. So this software actually solve this differential equation, which is academic use. You can download the INCA software and calculate the metabolic fluxes that is concentration dependent. But FBA as a whole cannot, it doesn't use the concentration. It assumes that the time derivative of the concentration is 0.
Thank you.
Right.
Very nice talk. I was curious about the virus model. You said you created a metabolic model for the virus? I didn't quite get that part. Could you elaborate?
Yes. So here you have the context-specific model, which are based on the RNA-seq data, but at the same time we have integrated the virus model inside the human gene, because the virus goes inside and become a part of the human metabolism. So you actually we have defined a virus biomass objective function for the entire network.
Biomass objective, not a metabolic reaction model,
yes.
Right?
It's a virus biomass objective function is there, and that you are optimizing using FBA.
Okay. Yeah, no, that makes sense. I was just wondering that
Yes.
did you create something else? Yeah, and you look at lung infected, the virus infected lung.
Yes.
And you create a context-specific model for that as well, right? Okay.
Yes.
And you said that. Did you see any differences in, how amino acids are taken up in these cases, or because you mentioned only fatty acids?
Yes. The amino acid part I have not discussed. The amino acids are also affected, and in terms of their formation and utilization.